All right, so this photosynthesis here, so this video is going to be one of the, if not the longest video in our AP Bio app, and on YouTube as well. So we're going to hop right in there. So we have 1, 2, 3, 4, 5, 6 questions. So these questions will show up again at the end, and that's a great time to make sure we can answer these. How does Calvin cycle work? Site of light-independent reactions. The photosystems, are we familiar with those and can talk about them in some detail? Um, Two things happen during light-dependent reactions. The process of non-cyclic photophosphorylation, and see if I can adjust this down. There we go. And how is ATP produced in light-dependent reactions? Okay, so we'll just hop right in here. Just start off with just the basics. Photosynthesis is a process by which chlorophyll-containing cells synthesize carbohydrate from carbon dioxide and water with the help of solar energy, and the byproduct is oxygen. It occurs in the chloroplast. During the light reaction, light energy is converted into chemical energy in the form of ATP and NADPH. This ATP and NADPH is used in the dark reaction to fix CO2 in the stroma. Carbon dioxide is one of the reactants needed for photosynthesis. It is not one of the products because it has to bind with RuBP in order for the Calvin cycle to function and to create sugar and oxygen. So we'll talk more about the Calvin cycle here in a few minutes. Light intensity, carbon dioxide concentration, and temperature all affect the rate at which photosynthesis occurs. Light is needed because it is the basis of photosynthesis. Without enough carbon dioxide, RuBP would be unable to combine with anything to work the Calvin cycle. In extreme cold or hot temperatures, photosynthesis may not function because there needs to be an optimal temperature for plant cells to be able to function. It is probable that there is a trace from ancient Earth where the atmosphere had no oxygen to compete with carbon dioxide to bind with RuBisCO. Therefore, RuBisCO is more inclined to bind with oxygen. Chlorophyll A is a specific form of chlorophyll used in oxygenic photosynthesis. It absorbs most energy from wavelengths of violet blue and orange red light. It also reflects green yellow light, and as such contributes to the observed green color of most plants. The photosynthetic pigment is essential for photosynthesis in eukaryotes, cyanobacteria, and prochlorophytes because of its role as a primary electron donor in the electron transport chain. Chlorophyll A also transfers resonance energy in the antenna complex, ending in the reaction center where specific chlorophylls P680 and P700 are located. Jumping over to chlorophyll B. Chlorophyll B is a form of chlorophyll. Chlorophyll B helps in photosynthesis by absorbing light energy. It is more soluble than chlorophyll A in polar solvents because of its carbonyl group. Its color is green and it primarily absorbs blue light. In land plants, the light harvesting antenna around photosystem 2 contain the majority of chlorophyll B. Therefore, in shade-adapted chloroplasts, which have an increased ratio of photosystem 2 to photosystem 1, there is a higher ratio of chlorophyll B than chlorophyll A. Carotenoids are a class of more than 750 naturally occurring pigments synthesized by plants, algae, and photosynthetic bacteria. These richly colored molecules are the sources of the yellow, orange, and red colors of many plants. Fruit and vegetables provide most of the 40 to 50 carotenoids found in the human diet. Chlorophyll A, chlorophyll B, and carotenoids are all pigments of photosynthesis. Chlorophyll A and B are green in color, and this hints as to why we see green when we look at a healthy plant. The green is reflected, which makes the color visible to humans. A pigment absorbs all colors it does not reflect. For example, chlorophyll reflects green, while it absorbs blue, red, violet, and so on. The color of a pigment depends on the color of the light that it reflects. The reflection of light is what makes color appear to humans. Antenna pigments don't help chlorophyll, and other pigments absorb light. They capture wavelengths of light that the other pigments cannot absorb in order to expand the spectrum of light that can be used for photosynthesis. Carotenoid and xanthophyll are yellow, orange, and red. The light harvesting complex, which is also called the antenna complex, is an array of protein and chlorophyll molecules embedded in the thylakoid membrane of plants, which transfer light energy to one chlorophyll A molecule at the reaction center of a photosystem. All right, so that was A and B. Let's jump to the photosystems real quick. Photosystems are what harvest light for the light-dependent reactions. There are two photosystems, photosystem 1 and photosystem 2. Photosystem 1 absorbs light at the 700 nanometer range, while photosystem 2 absorbs light at the 680 range. So to say here, on average, photosystem 1 absorbs light in the 700 nanometer range. It is meant to absorb light in this range in order to provide high energy electrons to produce NADPH and lower the levels of NADP. Photosystem 2 absorbs light at its range, which causes the split of water molecules in order to release oxygen. Photophosphorylation occurs in the thylakoids and involves both photosystem 1 and 2. A photosystem is a group of closely associated pigments, molecules that help in converting solar energy into chemical energy. Also referred to as light harvesting complex. It has two types, which are non-cyclic and cyclic. So we'll go over each of these. Non-cyclic first. The process of non-cyclic photophosphorylation starts at photosystem 2 and continues on to photosystem 1. It starts at photosystem 2 because that is where light is absorbed, and then it moves to photosystem 1 and ATP is produced. So some of the key points, both photosystem 1 and 2 are involved. Photosystem 1 and 2 are excited simultaneously with solar energy. Electrons are passed through the electron transport chain between photosystem 1 and 2. They are arranged in decreasing energy concentration. The electron acceptors are. Photolysis of water occurs. Oxygen is the byproduct and is responsible for the synthesis of both ATP and NADPH. Okay, moving on to the cyclic. Electrons in cyclic photophosphorylation travel from photosystem 2 to photosystem 1 and back to photosystem 2 in a cyclical motion. Only ATP is replenished during cyclic photophosphorylation. No NADPH is formed. This is because cyclic photophosphorylation only occurs when the Calvin cycle uses up all its ATP. Therefore, more ATP needs to be created. Some of the highlights for cyclic photophosphorylation: it occurs in the thylakoids and in stroma lamellae. They possess photosystem 1, photosystem 2, and NAD reductase enzyme is absent. Only photosystem 1 take part in cyclic photophosphorylation, only responsible for ATP formation, and there's no splitting of water molecules. Just a quick note on the photosystems 1 and 2. Photosystem 1 was named 1 since it was discovered before photosystem 2, but this does not represent the order of the electron flow. When photosystem 2 absorbs light, electrons in the In the reaction center, chlorophyll are excited to a higher energy level and are trapped by the primary electron acceptors. During light-dependent reactions, two major things happen. First off, light is absorbed and transferred to molecules of ATP. Second, water is split apart. The light is absorbed by photosystem 2 and photosystem 1 in the thylakoid membranes. Water is then split apart to replenish the missing electrons and protons. Light-dependent reactions use light to produce ATP, which then fuels the light-independent reactions. With photolysis, this is the name of the process that splits water apart. This process occurs in order to replenish the electrons that were lost in photosystem 2, as well as to provide protons to reduce NADP to the system. Electrons supplied by photolysis enter two ETCs, and that is where ATP and NADPH are formed due to chemiosmosis, which is what powers the production of ATP. So it's just, I think you're just going to see reactions, but this is the light-independent reactions. In light-independent reactions, ATP is produced as the flow of electrons through the electron transport chain is met with phosphorylation of ADP, which becomes ATP. This process then becomes photophosphorylation. Although the overall outcome of photosynthesis is to produce oxygen, the light-independent reactions produce sugar in order for the plant to keep functioning in the way it was intended to. Protons from water reduce NADP to NADPH2 in order for NADPH2 to carry hydrogen to the light-independent reactions. NADPH2 carries hydrogen to the light-independent reactions. The light-independent reactions use hydrogen carried by NADPH2 to make sugar. The stroma is the site of light-independent reactions, while the grana is the site of light-dependent reactions. Okay. Jumping to the theory of endosymbiosis. This is the unified and widely accepted theory of how organelles arose in organisms, differentiating prokaryotic and eukaryotic organisms. In endosymbiotic theory, consistent with general evolutionary theory, all organisms arose from a common ancestor. This ancestor probably resembled a bacteria or a prokaryote with a single strand of DNA surrounded by a plasma membrane. Throughout time, these bacteria diverged to form and function. Some bacteria acquired the ability to process energy from the environment in novel ways. Photosynthetic bacteria developed the pathways that enabled the production of sugar from sunlight. Other organisms developed novel ways to use this sugar. Oxidative phosphorylation produced ATP from the breakdown of sugar with oxygen. ATP could then be used to supply energy to other reactions in the cell. All right, we're jumping to the Calvin cycle. All right, so we've got the Calvin cycle and then photorespiration, and then we'll be done. All right, now this is a fairly lengthy video compared to what we usually have. The Calvin cycle uses the reducing powers of ATP and NADPH from the light-dependent reactions to produce sugars for the plant to use. These substrates are used in a series of reduction-oxidation reactions to produce sugars in a stepwise process. There is no direct reaction that converts CO2 to a sugar because all of the energy would be lost to heat. There are three phases to the light-independent reactions, collectively called the Calvin cycle, and they are carbon fixation, reduction reactions, and RuBP regeneration. PGAL, or G3P, is a 3-carbon sugar that is produced in the Calvin cycle. Carbon dioxide is combined with RuBP to produce two 3-carbon molecules. This is essential because the CO2 has to be able to combine with something in order for the Calvin cycle to function. A G3P molecule contains 3 fixed carbon atoms, so it takes 2 of them to create a 6-carbon glucose. It would then take 6 turns of the Calvin cycle, which is 18 ATP, to produce 1 molecule of glucose. The NADPH generated is used to reduce atmospheric CO2. It takes 2 G3Ps to build a 6-carbon glucose molecule. 6 NADP are converted to 6 NADP during the fixation step. Therefore, it would take 6 turns of the cycle, or 12 NADPH, to produce 1 molecule of glucose. And then finally, we are at photorespiration. Photorespiration is the process in which the enzyme RuBisCO makes RuBP, which causes energy from photosynthesis to be wasted. Photorespiration occurs when RuBisCO binds to oxygen rather than CO2. In photorespiration, 3 fixed carbon atoms are lost, while during the Calvin cycle, 6 fixed carbon atoms are gained. Photorespiration doesn't produce sugar because the RuBP binds to oxygen instead of carbon dioxide. Unlike regular cell respiration, no ATP is produced during photorespiration, and unlike regular photosynthesis, no sugar is produced during photorespiration. And finally, we get back to our questions. Okay, so this is the time for the pause button and see if you can answer these questions for yourself.